the lecture series in cell culture technology. So, today we are in the fourth lecture. So, in the last lecture I talked to you about the challenges of developing a neuromuscular junction. There are two challenges what we put forward. Week 8, lecture 4. So, these are the two challenges. How to integrate this system on a microcantilever assembly was one, and the other challenge was how to co-culture a muscle and a neuron cell type to study neuromuscular junction. So, these two problems comes with another additional issue, that you wanted to have a defined medium which is a serum free, and it should be a common medium. This was very challenging from the word go, because when you have to grow the muscle, try to realize how the in vitro systems can become so very daunting for to work with, and try to appreciate that how you can bypass all these things so neatly. So, when you talk about muscle, if you remember, the first thing what we told you is, this is the picture right out here. So, these are the muscle cells which divide. Post division they form this kind of alignment, and then of course, so this is the phase of, I told you about the differentiation. So, these are your muscle cells you are plating in a dish. Okay. Your initial plating, these are muscle cells. Post plating, you have medium which will promote their division initially first 3-4 days. You are allowing them to divide. Post division, they will align like this. They will be elongated, they will be aligning like this, and soon after that they will lose their individual identity and they become a tube like this. Now, in this process, this is which is the division phase, this is which is the differentiation phase. Differentiation phase. Now, in order to divide, so you have to use molecules which allows the division, and then you have to, so I am just putting the dividing molecules, then you have to withdraw the minus sign showing the D molecules and allow the cells to grow in D prime, D double prime, which is in the milieu of differentiation molecules. Okay. So, this journey becomes complicated when you are adding into the system another component which is a neuron which never divides. So, here you have the, in this case it is a motor neuron which do not divide. Now, whenever you are using anything which these kind of molecules which are mostly basic fibroblast growth factors which are used, and this medium, this whole thing has to be a common medium. This has to be a common medium and it should be serum free. So, the way we achieved it took us a while to achieve this interesting feat, but we eventually did achieve it. So, what we did first is that we took the muscle, as I have mentioned in the previous class, and we did a first level where the muscles are contaminated with fibroblast and all. We just allow the muscle to sit there and the fibroblast kind of settle down and the muscle cells were in the top. Okay. What you do that you take out this suspension, okay, and you take out the suspension, so you have the more or less the pure myocytes sitting out there. Then what you do, you plate these myocytes on a dish. So, once you plate these myocytes, you tweak around with the basic fibroblast growth factor concentration. You use very, very low concentration. Of course, that has its own problem because then the cells won't divide in sufficient number, but that is one degree of compromise you have to do. So, you plate the cells and on top of it you put the medium. So, what you have in a dish is something like this. So, here you have the cell culture dish where you are growing them. So, you have these muscle cells which are kind of settled on the surface after panning. I told you how you are removing the fibroblast because the fibroblast will be settling down faster. So, you have the muscle cells out there. Then you add the medium with FGF, and there are other components. In the handout I will give you, you can have a look at it, what are the other components which are being used. Now, you leave this in a dish for approximately 1 to 2 hours. It is during this time most of these cells will, first of all, of course, most of these different growth components will bind on the surface of the cells and will initiate the necessary reactions what they have to do. This red color is showing all the different kind of binding of FGF and other growth factors which are being used, and simultaneously these cells will start to secrete their own extracellular matrix. Okay. Now, after 1 hour, while still the culture is 1 to 2 hours, what you do, you remove a small part of the medium, very small part of the medium, good enough that it just mildly cover the top, and then you introduce your motor neurons. They will be sitting something like this, and these motor neuron mostly comes from, in case of rat, in case of fetal culture, they come from E14 rat embryo, and they are coming from ventral horn of spinal cord, whereas your skeletal muscle for the cultures are coming from E18 rat fetus for hind limb skeletal muscle. So, that is where you are getting the myotubes. So, after 1 to 2 hours you add these motor neurons. Then of course, the medium what you have removed initially, you replace it back, because you have to realize in the very early phase the cells secrete so many important growth factors, so many, it is a plethora of growth factors they secrete, which are extremely essential, extremely essential. and while you are putting the medium back, you add some fresh medium on top of it, which is without basic FGF. So, what you essentially did, you actually in this whole process, you diluted the effect of basic FGF. Okay. And if you can hold this culture for, I would say, anything between 6 to 10 days and slightly longer, say 15 days, you will see wonderful neuromuscular junctions getting formed like this. And one of the most beautiful tests you can do is, there are a couple of tests you can do. One test is, say for example, you see the motor neuron out there, you stimulate the motor neuron with an electrode, you should be able to see with a time delay, you should be able to see a contraction taking place in the muscle, or you should be also able to see parallelly. And if this is E2, this is electrode 1, and what will you see is 2 scale, this is showing for E1 and this is E2. So, here you are giving the impulse at E1, and you see there is an electrical activity generated out here, and then with a delay you will see another electrical activity happening in the muscle out here, and simultaneously you will see a mechanical motion or a contraction happening at the zone. So, this is your first impulse coming through. So, this is the stimulus. Say for example, you needed a stimulus. This is the first impulse. This is an electrical activity of the neuron, followed by out here the electrical activity of the muscle, and simultaneously the mechanical activity of the muscle. You really can physically see it. Apart from it, there is another way you can evaluate it, is that out here you can map the NMJ or neuromuscular junction with specific protein probes or antibodies. Probes or antibodies. Simultaneously, the antibodies could be, you can probe them for, I would say, against acetylcholine receptor, because there are a lot of acetylcholine receptors which are present on the muscle. You can probe them with bungarotoxin. So, bungarotoxin is a toxin which will block the transmission in the neuromuscular junction. So, you can parallelly do one more thing. You suppose given a stimulation here and you add something, there is a drug called curare, which is used by the tribes in Amazon basin to hunt down the deers or other animals. What it does is, curare upon binding, it paralyzes the neuromuscular junction. So, if you add curare and if you give a stimulus, you can have a stimulus, but you will not see any signal out here. Your signal will be missing out here, and there will not be any motion out here. This is the effect of curare. Curare can block the neuromuscular junction in a very unique way. It's a, by the way, it's a reversible blocker. After a while, curare's blockage stopped. So you can actually use these kind of things. And not only that, if you are seeing that this whole setup is spontaneously contracting, you can stop the contraction by adding curare. You will see upon addition of curare, there will be shaking like this, and then the muscle will stop. So all these things can be seen in vitro, and these are proven track, as I will hand over all the papers to you. These can be seen very neatly and they could be evaluated. But our problem was even much more challenging. We wanted to do this on top of a cantilever. This is what we are kind of looking forward to. So in order to do that, what we needed was a common medium and a different plating rules. So the plating rule was, or is, you plate the muscle first and wait for 1 to 2 hours and then add the motor neuron. And I told you the source of motor neuron, which is from the E14 ventral horn of the rat embryo, and skeletal muscle you are collecting from E18 rat fetus. Okay? So if you can really pull it through, as of now, to the best of my knowledge, as of now, no one has been successful in achieving this kind of such a neat system. Muscle has been done, but really integrating it with the neuron still will take a little bit more time, because this is not an easy system to work with. But these are some of the cleanest systems which in years to come is going to change the face of cell culture or traditional cell cultures, what people have been following for years together. Like these are the modern day technology where we are ushering into, where cell culture is no more a territorial dominance of the biologist. It is now a very integrated journey of engineers, mechanical engineers, chemical engineers, electrical engineers, and designers, biomedical scientists, biologists, physicists, chemists. So in a way, you have to have a fairly good idea about cell patterning. You should be able to know the different kind of thin layer technology, thin layer technology, another area, and integrating electrode into such structures. So as of now, this approach also, like on a cantilever, say for example, here you have a cantilever having embedded electrode or embedded electrode systems. These are not easy job. You are doing at a micron level, and these are still, it will take a little bit more time. Technology is there, but really to make it happen the way it is not something so easy. Okay, so in this segment, what we are trying to do and is kind of exposing you to those technologies which are still not part of textbook. These are futuristic technologies, but these are the technologies which will drive the future. Say for example, you wanted to test a system for ALS, amyotrophic lateral sclerosis. That is a situation when motor neuron is unable to communicate with the muscle because they die out. To create a system, how you can do it? These are the system. You want to study Parkinson, and you want to have 3 layers. Say for example, a motor neuron coming from substantia nigra, a motor neuron in the spinal cord, and here is the muscle. So the signal which is missing is here. Substantia nigra neuron is not sending a signal. How one can really study, make a Parkinson model on a dish, or how you could have the cell patterning technology to make circuits like this, where you can study Alzheimer's. And in one of the small modules, end of it, I will talk about the microchannel technology, where we will talk about how this microchannel technology could come very handy to study single neuron to single neuron communication within hippocampus, which could throw a lot of light into our understanding or the deep understanding of the signals generated by the hippocampal neuron while cross-talking with each other. So, I will close in here. So, in the last class in this series, we will talk about the electrical excitability and how we achieve it. Okay? Thank you.